Hello guys, so in this chapter we're covering everything about the cell. In previous chapter we were able to discuss all the different macromolecules, including carbohydrates, lipids, nucleic acids, and also proteins, right? So we mentioned that carbohydrates are very important for the cell in order to give us energy. They're also very important components of bigger, more complex molecules like nucleic acids. So we know that nucleic acids are very important for the storage of genetic information, and they are like this cookbook of life that have all of these amazing recipes to create proteins. We also know that lipids are very important to give us that plasma membrane as part of the cell and be able to hold and keep everything inside of the cell in that compartment and keeping it safe away from that environment surrounding the cell. And we also know that proteins are pretty much the building blocks of life, and all of these different macromolecules are made out of carbon, right? So now that we pass over those macromolecules and understanding each one of them one by one, we're not going to be able to put everything together and talk about their role in the cell as a whole. So before getting into each one of the components and organelles of the cell, we have to make a stop and make a difference in understanding the difference between prokaryotic cells, and we also have eukaryotic cells. So first, let's make like big major differences when we have between them. So number one, we know that eukaryotic cells versus prokaryotic cells, there's going to be one big difference between them, right? And that is going to be the presence of the nucleus, right? So eukaryotic cells have a nucleus, and that is very important because in the nucleus we are able to store all of that DNA, all of that genetic information that holds all the recipe of life, right? And to create new proteins and to create new cells. So in eukaryotic cells, you're going to see that we have that nucleus. So that nucleus is keeping safe, right? Like in a drawer and very locked with a key and stored right there to have all that genetic material and to keep it safe. Now, versus prokaryotic cells, you can see in prokaryotic cells, we don't have the presence of a nucleus. So the chromosome pretty much is going to be one big chromosome, right? Pretty much floating around all the cytosol in that bacteria, in that prokaryotic cell. So it's going to be pretty much everywhere. Talking about the chromosome and the genetic material in prokaryotic cells, another difference regarding that is going to be that prokaryotic cells, they're going to have also these plasmids, and plasmids are going to be these little gene cassettes that are floating around as part of the prokaryotic cells. Now, it is very important to make a difference between the main chromosome of the bacteria and also of all of these plasmids, which are little gene cassettes, right, of genetic information from the environment. All this big chromosome is going to have everything, all the genetic information regarding the components to create the bacteria, the structure, the cell wall, what type of cell wall it has, when it's going to go through replication in a binary fission style, pretty much everything that is needed for the bacteria as part of their structure and genetic information. But when we have all of these plasmids or gene cassettes, these are very important circular genetic information that bacteria are able to get from the environment. So, for example, if that bacteria notices that it's in an environment where there is a lot of genetic antibiotic resistance, where there's a lot of maybe ampicillin or amoxicillin in that environment, this bacteria is going to go ahead and take little chunks, all of these little plasmid cassettes that comfort and gives it that antibiotic resistance. So we can see the process of evolution and natural selection happening really fast, right, through the problem of antibiotic resistance that we have right now. So those plasmids will always reflect kind of like the environment that bacteria is currently present on. So can see it in the terms of pollution, antibiotic resistance. Some of these bacteria are even have the ability to have that against metals, because sometimes there's a lot of metals and they use those metals to comfort also a little that extra layer of protection. So I love seeing the problem antibiotic resistance because that's where we are able to see natural selection happening like in a very quick process, right? So we have prokaryotics. So remember, we don't have a nucleus. All the chromosome is going to be all there floating around in that cytosol of the bacteria. We have the little plasmids, the little gene cassettes. I don't think you can have them here labeled, but just like circular gene cassettes that are part of that genetic material, and genetic material, and they're part of that resistance part or a defense mechanism for that prokaryotic cells. Okay, so we have one big information and one big difference so far talked about the eukaryotics and the prokaryotes in the presence of the nucleus holding the DNA and the presence of no nucleus, right? Pretty much chromosome all over there. Okay, another big difference that we can tell from eukaryotic cells and prokaryotes, you're going to see that prokaryotes have a cell wall. So let me go ahead and select it. It's over here. It's the cell wall, right? So the cell wall is going to be an extra, very thick in some bacteria, a very thick layer made out of glycoproteins or glycolipids, sometimes of these peptidoglycans, right? So it's going to be very thick in some bacteria. And in the lab, we have a process that we go through to make difference when we're taking like a sample to make a difference of what type of bacteria are we dealing with. And that's like a big way to make to label it and to make the classification of the type of bacteria. And that's if the bacteria is gram-positive or gram-negative. And we are able to detect, to make that classification because of the cell wall. So for example, in the case of Staphylococcus aureus, right, or like gram-positive bacteria, Staph is a type of gram-positive bacteria. These bacteria have like a very, very, very thick peptidoglycan wall. So you can pretty much be able to detect it. So they have a very thick layer of protection, that Staph, right? So in comparison to gram-negative bacteria, for example, gram-negative bacteria, they do not have thick cell wall. They do have cell wall, but it's going to be very thin, right? It's just going to be like minimal. But however, for example, gram-negative bacteria like E. coli that causes a lot of GI infections, right? Has other mechanisms that has been able to develop as part of that resistance and kind of like to be a very infectious strain. For example, one of them is going to be one of those plasmids, right, that confer them that extra antibiotic resistance. So if you're sick and take a course of antibiotics and it doesn't work, pretty much because that bacteria has an extra plasmids and extra layers of protection. So, okay, you can see that it has a cell wall. Eukaryotic cells do not have that cell wall, okay? They do have the plasma membrane, that double phospholipid bilayer made out of phospholipids, right? That is able to confer that protection within the cell wall and be able to hold all of those organelles in place, right? And protect them from the environment. But we have a plasma membrane. So remember, plasma membrane, no cell wall. In the cell wall, we are able to have, in the prokaryotic cells, we do have a plasma membrane, but that plasma membrane is going to be surrounded as well by that thick layer of a cell wall. So that's going to be another difference, right? Another thing is you're going to see that in both of them, that's something that is similar in both. Prokaryotes have a lot of these ribosomes. You're going to see all of those little green circles. They're supposed to be these ribosomes. They have a lot of ribosomes. In ribosomes, yes, we also have in eukaryotic cells ribosomes, right? The only difference between them, it might be like the size of the ribosomes in both eukaryotic cells and prokaryote. Not so much a big difference, but there is a difference in size. So that might be like the difference between the type of ribosomes that we have between prokaryotic cells and eukaryotic cells. But yeah, both of these type of cells have ribosomes because they're very important in the building of proteins, right? Another thing, you're going to see that in prokaryotes, we also have flagella. You're going to see this long little tail that it has. That's going to be the flagellum. So the flagellum assists in prokaryotic cells to move, to give them movement, right? So there are these long projections. They move like in a whip-like style but it allows the prokaryotic cells to move in that environment. Some eukaryotic cells, for example, like sperm cells, do have the presence of a flagellum, and they move like in the same whip-like style, and that confer movement to them. Another difference is going to be the presence of pilus, right, of pili, and these, I cannot point, it's kind of like difficult for me to point things. There we go, the pilus, right? So these pilus or pili, they're like needle-like projections, but it helps the bacteria kind of like to attach to other surfaces. For example, some viruses or some bacteria, they're able to hang on to the respiratory system, right? It allows for it to pretty much to hang on, to cling into it, and be able to deposit or deliver all of its infectious particles and produce an infection, right? So that will be kind of like another difference. Let me see. In eukaryotic cells, we have like more complex organelles. We have the rough endoplasmic reticulum, right? Because you're going to see that in prokaryotes, we have the ribosomes right there, but we have the rough endoplasmic reticulum right here in eukaryotic cells that help us with the folding and the shipping and the processing of all of these, the different proteins and the different structures and shapes of the proteins. We also have the Golgi apparatus that we're going to talk a little bit more in detail in a little bit. We also have mitochondria. Mitochondria help us with the making of ATP, right, and transforming all these macromolecules like glucose and be able to break it down into little different molecules and change the shape and the organization of glucose. So finally, it can enter the mitochondria and be able to produce all that ATP that we need, right, in our bodies. So we have like big difference. We also have lysosomes, for example. Lysosomes are very important recycling centers, right? That if they detect some molecules or some particles that might be damaging to the cell, or maybe they are not functioning very well, they're able to release some acid enzymes that pretty much destroy and break away these proteins or these molecules and get rid of them, sometimes recycle them. What other things do we have? We have many organelles. We're going to be talking about them. I just wanted to give like a brief introduction of the main differences between prokaryotic cells and eukaryotic cells, okay? All right, guys, so that's it. If I forgot something else, I'll go ahead and make sure to write it down, okay? Or go ahead and type down what are other differences am I missing that we discuss in class from prokaryotic cells and eukaryotic cells. All right, let's move on.